So, dear students, you are welcome in this class. For next 20 or 23 minutes, I would try to introduce you to the essential plant nutrients. Actually, what happens when you see the composition of the plant? If you take any plant biomass, plant material which is growing on the earth, and if you remove the carbon, hydrogen, oxygen, if you oxidize it, if you burn it, whatsoever way, or if you digest it, and you need to remove carbon, hydrogen, oxygen, and whatever is left, that that is called as ash, ash. So ash contains more than 60 elements. In nature there are more than 110 elements which are natural. So your plant ash will also contain 60 elements. 60 elements are there. That means the plant physiologists and people started thinking from very beginning. For say 200 years before, they started thinking that what it is made of, this ash. Then they found that, okay, this is made up of 60 elements. So then people started thinking that all these 60 elements essential for the plant growth, because they thought that because they are there in the plant, so are all essential or not? Then people started thinking, and in the end they come out that no, it is not maybe 15, maybe 16, maybe only they reached 17 essential nutrients. So now we understand that out of 60, which are part of the plant ash or which are there in the plant, only 17 are essential, without which plant cannot grow. Plant can grow without silicon, plant can grow without vanadium, although they are part of the plant ash. So therefore the concept of this essentiality started. So I think you know chlorosis is the greening of the plant. When a plant becomes green, loses color, loses chlorophyll, then it is chlorosis. Necrosis is the death of the plant tissues. This necrosis can happen due to microbial attack, and necrosis can also happen due to deficiency or toxicity of the elements or nutrients. Necrosis can also be because of nutritional disorders. A nutrient is a nutritive substance or ingredient such as the major and minor elements necessary for growth and development of the plant. Nutrient antagonism, the suppressing effect caused by one or more plant nutrients on the uptake and availability of another nutrient. That is your nutrient antagonism. It can happen in soil as well as in the plant system at cellular level also. And nutrient availability is the extent to which one nutrient or nutrients are available for plant use. Nutrient deficiency, when there is shortage of nutrient in the soil or in plant, that is your nutrient deficiency. And nutrient stress occurs when the quantity of nutrient available reduces the plant growth. It can be from either a deficient or toxic. So nutrient stress can be because of deficiency or toxicity. Likewise, water stress. Water stress is not always due to shortage of water or due to drought. Water stress can be because of excess water also. Waterlogging condition is also water stress. Similarly, if you have higher nutrients, too high levels of nutrient, or too low levels of nutrient, that is also your nutrient stress. Then essential plant nutrients. As I talked in the beginning, that which nutrient has to be essential. Then Annan and Stout 1939 described the criteria for essentiality of nutrient. If a nutrient or if an element is able to qualify these three criteria, then it will be called as essential. Otherwise, it will not be essential for plants. So more or less, these three criteria are still valid. People respect them, they accept it. With few exceptions may be there, but you need something, you need some criteria. And no other alternative criteria has come up again after Arnan and Stout. If we don't have alternative, that means this is still valid, this Arnan and Stout criteria. So what did they say? A deficiency of an element makes it impossible to a plant to complete its life cycle. So life cycle, you know, for plants, you have lower plants, higher plants, and different classification of the plants. So they have different phases in their growth, like germination, then growth phase, then reproductive phase, then maturation. In general, these are the growth phases of plants. if they are able to complete the life cycle, passing through all these growth phases, then that nutrient will be essential. In the absence of a particular nutrient, if plant is not able to complete its life cycle, then it will be essential nutrient. Second is that the deficiency is specific for the element in question. Means requirement of a particular nutrient can be met only by that element. It cannot be substituted or replaced by other element or other nutrient. For example, you get function of say function of nitrogen. So function of nitrogen means making of chlorophyll or RNA, DNA can only be by nitrogen only. Other elements cannot substitute functions of this nitrogen. Similarly, however, some people say that exceptions are there. Some role of calcium, magnesium, and potassium can be met by sodium. All these four elements are brothers. This calcium, magnesium, and potassium and sodium. Sometimes they can substitute each other's function. Potassium is happy to substitute sodium, and sodium is happy to substitute function of potassium. So sometimes it happens. But still, up to some extent or up to full extent, you can say this criteria is still valid with few exceptions of calcium, magnesium, potassium, and sodium. The element is directly involved in the nutrition of the plant. For example, as a constituent of an essential metabolic or required for the action of an enzyme system. They must be directly involved in the metabolic activity. In plants, you get two kinds of metabolic activity. One is photosynthesis, other is respiration. Anabolism, catabolism. So in these, you need certain biochemicals, certain molecules in the plant. You need chlorophyll, you need other molecules in the plant system, you need glucose and so on. So in the synthesis or manufacture of this, they have to be directly involved. They have to be part of those molecules, or they are necessary for conduct of synthesis or for synthesis of several constituents in the cells. Their presence is necessary. Somewhere they will act as, say, catalyst, or they will act as enzyme or so on. So the essentiality of most micronutrients for higher plants was established between 1922 to 1954. So it actually started with carbon, hydrogen, oxygen, and then nitrogen, and then followed by micronutrients. The essentiality of nickel was established only in 1987 by Brown and co-workers, and list is not final. Who knows, in 10 days, 10 years, and any other element can become essential, who knows? But as on today, there are only 17 nutrients which are essential for plant growth, and nickel's essentiality was tested in barley. In barley, for the germination of barley seed, nickel was found out to be essential. Or for viability of the barley seed, nickel was essential. If there is no nickel, barley seeds will not be viable. Therefore, it was proved that it is essential for all other plants also. You can see the discovery of essentiality. Essentiality, who established the essentiality of different nutrients? So you can see carbon, hydrogen, oxygen, nitrogen. It was de Saussure, I don't know how to read it, de Saussure, 1804, phosphorus, Ville, 1860, sulfur, Von, Sach, Knopp, and potassium, calcium, magnesium, RL, also Knopp, 1860, manganese, McHargue, 1922, copper. So you can go through it, Lippmann, Sommer and Lippmann for zinc, chloride for Brower et al., Nickel Brown et al. So these are the discoverers of essentiality. There may be some confusion in these also, but some are sure, like boron. There is no confusion. It is Barrington always. Barrington 1923. So some quiz exam, it is important. See this slide. This is very important slide where you can see the essentiality of mineral elements for higher and lower plants. You have two kinds of plant. One are higher plants, lower plants. Lower plants you can consider fungi, algae, bacteria. So in this case, you can see macronutrient, nitrogen, phosphorus, sulfur, potassium, magnesium, calcium. These nutrients are macronutrients. and they are essential, all are essential for higher plants. But calcium is not essential for fungi. You can remember this. Calcium is not essential for fungi. In micronutrient, you have iron, manganese, zinc, copper, boron, molybdenum, chloride, and nickel. All are essential for higher plants, but boron is not essential for higher plants. So from this, you can remember that calcium and boron, they are not essential nutrients for fungi. And however, there are certain beneficial elements like sodium, silicon, cobalt, iridium, vanadium. This silicon has very important beneficial role in cereals, and cobalt has very important role in legumes. And this sodium has very important role in sugar beet and sugarcane. And now, if you see the classification of essential plant nutrients, there are two groups. I hope you are able to see this slide. There are two groups. One is non-mineral. Non-mineral, see, they are non-mineral or framework elements. They are taken from air and water, 3 number, carbon, hydrogen, oxygen. Then mineral elements or mineral nutrients taken up from soil in ionic form. Now you also understand they are taken in organic form. There are 14 in numbers, macronutrients, micronutrients, and there is no any limit. Some people say for macronutrients, it is more than 0.2% concentration like this. For micronutrient, less than 1000 ppm. There is no such criteria. If you want to define them, the nutrients which are required relatively in higher amounts are macronutrients, and nutrients which are relatively required in low amounts are your micronutrients. So sometimes students get confused that it is, if it is less than so much ppm, it is micronutrient. There is no such criteria. Then macronutrients are further divided upon into primary and secondary nutrients. Here also you see relative concentration or uptake of NPK is more than calcium, magnesium, and sulfur. So therefore they are called primary nutrients, and then calcium, magnesium, sulfur, they are secondary nutrients. But there is other purpose also calling them primary nutrients, because they are mostly in commercial fertilizer. Farmers also give them primary importance, and they are usually deficient in soils, nitrogen, phosphorus, sulfur. So therefore, they are of prime importance. From that angle also, they are considered as primary nutrients, not from the point of view of the concentration alone. Their uptake requirement is high, therefore they are primary. Their deficiency is also high in soil. Therefore, application of these nutrients is of prime importance to the farmer. Therefore, they are primary nutrients. Otherwise, you can also add your views why they are called primary nutrients. Secondary nutrients are in no way they are secondary, but the farmers give preference to them once your primary nutrients are applied. And then the secondary importance is given to secondary nutrients. So far, concentration and uptake is concerned, the sulfur and phosphorus, they are brothers or they are sisters, you can say. Phosphorus and sulfur are sisters. They behave almost same. Their uptake and concentration in different part is almost similar. You can remember this point. Phosphorus and sulfur, they are brothers, they are sisters. I'm not saying that they will have exact concentration in all the plants part and so on, but almost their concentration and uptake is almost near, and their behavior and many functions at the plant level are also quite similar. And coming to micronutrients, they are zinc, copper, manganese, iron, boron, molybdenum, chlorine, and nickel. So they are 8 in number. So in total, you get 17 essential elements, which are essential. Now you see ionic form. I have already shown you ionic form of these nutrients. You know it. And then framework element. Let us discuss a little about framework element, carbon and oxygen. They can come from carbon dioxide. You see, hydrogen from water. Oxygen is also there in the water. And these 3 elements required in large quantities for production of cellulose, starch. You know, all these organic molecules, compounds are made up of carbon, hydrogen, oxygen. Your carbohydrates, your fats, your lipids, all need carbon, hydrogen, and oxygen. So it makes up 95% of the plant biomass, carbon, hydrogen, oxygen, and remaining makes up 5% only. Means 5% is your ash or your minerals, and remaining 95%. This is again very average, very casual value. This will vary with the plant, with the biomass, kind of biomass, climate, environment, soil. So many factors will affect. This is very average value, 95% carbon, hydrogen, oxygen. And in this 95, 40 to 42% carbon, 40 to 42% oxygen, and 5 to 8% or 5 to 6% hydrogen. Remaining 5% is your minerals. So 14 elements are mineral nutrients taken in minerals forms, and you got macronutrient, micronutrient as per their relative amount required. Relative amount you need to write. The difference in plant concentration between macronutrients and micronutrients is enormous, enormous difference. The relative content of nitrogen and molybdenum in plants is in the ratio of 10,000 into 1. See, nitrogen atoms and your molybdenum atoms in the plant system, their ratio is 10,000:1. So see the difference in macronutrient and micronutrient. Plants need about 40 times more magnesium than iron. So you can find the differences in relative concentration of nutrients. You see this data from Epstein 1965 and 2005. He written very excellent book on mineral nutrition. So you can see nitrogen is the element, symbol is N, and the concentration in plant is 15,000 ppm, 15,000 milligram per kg, which is about 1.5%. These are average values given by Epstein, and you can see relative number of atoms. If you take it one for nickel, if you consider one atom for nickel, or one atom for molybdenum, then nitrogen atoms are 10 to the power 6. Then potassium atoms are 250,000. Calcium, so you can also see the percentage or their concentration. So please study this table, and you can see potassium is 1%, calcium 0.5%, magnesium 0.2%, phosphorus 0.2%, and sulfur 0.1%. So likewise, you can see micronutrient concentration is very, very low. So you can see here. Now we discuss macronutrients are 6 in number: NPK, calcium, magnesium, sulfur. NPK are often referred to as primary nutrients. And why they are primary nutrients? Because they are most common elements found in commercial fertilizer. So this is one definition for primary nutrients, that because they are most common elements found in commercial fertilizers. Calcium, magnesium, sulfur are referred to as secondary nutrients and are also found in fertilizers and amendments, but they are not largely applied by farmers. It is NPK which are largely applied by farmers. Micronutrients are 8 in number. You have already seen the list. Micronutrients are required in relatively minute quantity. So this is how you need to define the micronutrients. So these are the these are the elements which are essential for plant growth, and plants cannot survive by in the absence of micronutrients, but they are required in relatively minute quantities and rarely limit plant growth in the environment, because micronutrients requirement is too low and most of them are present in the soil. So therefore, rarely limit the plant growth. Of course, now I've already shown you data about our soil fertility. Some soils are getting deficient in many micronutrients. But see the case of nickel, we never experience. See the case of chloride, we never experience deficiency of chloride in soils. Or see case of copper, normally not there. In some soils it may be there. So micronutrient, cobalt, sodium, vanadium, and silicon are sometimes called as beneficial plant nutrients. They are not required, but they benefit the plants. Cobalt is required for nitrogen fixation, so it is good for legumes. Silicon found in plant cell walls and produces tougher cells, increases the resistance of plants to piercing and sucking insects, and decreases the spread of fungal diseases. If you see any grassy plant, say sugarcane or rice or wheat or any plant, and take the leaf and see the edges. If you take your finger like this, take your finger and rub the edge in the opposite direction, edge of the leaf, it can cut your finger. The edges are very sharp. Edges of sugarcane are very sharp. If you rub your finger, if you put that leaf this way in opposite direction, it will cut your finger. That is very sharp, and it is because of presence of silicon in those areas. So silicon found in plant cell walls and produce tougher cells, increases the resistance of plants to piercing and sucking insects, and decreases the spread of fungal diseases. So silicon has very important role, though it is not essential, but it is beneficial for cereals. Now see this Mengel and Kirkby. Mengel and Kirkby, they have divided, they have made a classification based on biochemical and physiological functions in plant. So one classification of plant nutrient or essential plant nutrient was based upon their relative requirement. Relative requirement means you have framework element, you have mineral elements, then in mineral you have primary, secondary and micronutrients, and then this primary NPK. So you know that classification. That classification is there, yet you find another classification. So you can also use this classification to classify the nutrients, and this was suggested by Mengel and Kirkby 2001. These people have been very important workers in plant nutrition, and they have written one book, which was just like a Bible for people who used to study plant nutrition. Now, this book is not used by many people these days, but it was very good book in its time. So groups are 1, 2, 3, 4. Group 1 includes your carbon, hydrogen, oxygen, nitrogen, and sulfur. So based on biochemical and physiological functions in the plant. Major constituent of organic materials, this carbon, hydrogen, oxygen, nitrogen, sulfur. They are in one group, constituent of organic material, essential element of atomic groups which are involved in enzymatic processes. Then second group is phosphorus and boron, esterification with native plant alcohol. Phosphate esters are involved in energy transfer. Then third group includes your potassium, magnesium, calcium, manganese, and chloride. Non-specific functions establishing osmotic potential. Then fourth is iron, copper, zinc, molybdenum, present predominantly in a chelated form in prosthetic group, enable electron transport by valency chain. So actually what happens, this group is particularly important, this classification for students. Sometimes if a question is asked, write down the classification of essential plant nutrients. Almost 100% student will give their answer, the first classification, macronutrient, micronutrient, primary, secondary, and so on. But if your answer also gives this classification, then you will get more marks, I'm sure. You will get better marks than those who are giving just one classification. So that way your answer will become much better than the other people. So this is how you can use this. Then primary nutrients, NPK are often referred to as primary nutrients. The most common elements found in commercial fertilizer. This is discussed, I think. Frequently managed by addition of fertilizer into soil, while others are usually found in sufficient quantity in most soils and no amendments are usually used. Now you can see the primary nutrients are usually lacking in soil because plants use them in large amount for their growth and survival. So you can see this is relative uptake of NPK in rice producing 5 tons. So you can see nitrogen uptake is on an average 120 kg, and see the potassium uptake. It is rather more. In cereals in general remove more potassium in the form of K than N, nitrogen. Of course, phosphorus uptake is less. This is 20 kg phosphorus in the form of P. So you can see And if you see micronutrients, in 1 hectare, micronutrient like your zinc is removed about half kg, 400 to 500 grams. Zinc is removed in 1 hectare area. So you can see, and similarly, iron may be 1 kg, 1.5 kg, not more than this in 1 hectare crop of cereals. So you can see relatively this macronutrient or NPK, primary nutrient uptake is very high from the soil. Now you see deficiency and toxicity symptoms of nutrients. And I think half of the slides are left. I can finish it here and start tomorrow from this point onward. Okay, is it okay? Now your time is also over.
Okay, sir.
I stop here. I feel tired now. So dear students, good morning to all of you. I'm very pleased to be with you in this lecture. And today is 5th September 2022. And all of you wanted to wish us a very happy Teachers' Day. And I receive your greetings and wish you good luck, all the best. Now we start the class. Yesterday we started introduction to essential plant nutrients, and we tried to define what is meant by essential plant nutrients. Means plants cannot complete their life cycle in the absence of that particular nutrient, and nutrients are specific. The need of one nutrient cannot be replaced by another nutrient, and they play direct role in the plant metabolism. So there are 3 criteria. If an element is able to qualify all these 3 criteria, then it becomes essential. So we have seen this, that plant is not able to complete its life cycle in the absence of that particular nutrient. Deficiency is specific and they play direct role in plant metabolism, that is anabolism and catabolism. And we have seen some history of the nutrients, who discovered the essentiality of different nutrients. And we have also seen that calcium and boron, they are not essential for the growth of lower plants like fungi. And we have seen simple classification of non-mineral and of framework element and then mineral elements. Mineral elements are macronutrient, micronutrient, and then macro are primary, secondary, and you have 8 micronutrients and different ionic forms in which nutrients are taken up by plants. And we have seen the importance of carbon, hydrogen, oxygen. They make the main part of the plant body, that is approximately 95%, and 5% are minerals. Overall, you can see these 14 elements are mineral elements, and we have seen the relative concentration of different mineral nutrient in the plant bodies. So you can see nitrogen is on an average, this is across the all terrestrial plants, 1.5% is the mean nitrogen percentage. And you can see the relative number of atoms. They are 10 to the power 6 for nitrogen. So molybdenum has just 1 atom. So you can see the relative concentration of different elements in the plant system. On an average, 1 hectare crop of rice removes about 400 to 500 gram of zinc and 1 kg of iron. So you can see very little quantity is removed. But if you see the case of nitrogen, at least 120, 130 kilogram nitrogen and 140 kilogram approximately, or 130 kilogram potassium is removed from the soil to produce 1 hectare cereal crop. So you can see macronutrients are primary and secondary nutrients and micronutrients. And we have seen some beneficial nutrients like cobalt, sodium, vanadium, silicon, etc. They have their importance. In addition, they are not essential, but they help in the better growth of the plants. We have seen yet another classification by Mengel and Kirkby. He has divided essential nutrients into 4 groups. And then we have seen the relative uptake. If N uptake is say roughly 120 kg nitrogen per hectare for 5 ton per hectare rice yield, then potassium uptake is 130 and phosphorus uptake is 20. So you can see, take an idea that to produce 1 ton of rice, at least 24 kilogram nitrogen is taken up by plant. Now, this is the starting for today. Deficiency, toxicity, and role of essential nutrients that we will discuss in very brief. But before that, please try to understand this picture. This is by Fink, 1992. Plant growth and yield dependence on nutrients. Plant, yes. Yes, you have any issue? So plant growth and yield dependence on nutrient supply. So please try to understand this picture critically. You can see on x-axis, this is x-axis on the bottom, and this is your growth factor or independent variable. So mic is on. Somebody's mic is on. Please put off your mic. So you can see on x-axis, it is growth factor, or you can say it is independent variable. And on y-axis, it is dependent variable. Dependent variable may be here, it is growth, in terms of growth or in terms of yield. You can take either of these. And x-axis, it is the nutrient concentration. Nutrient concentration in soil or in plant. You can see both are written here, plant and soil. So nutrient concentration in plant or soil is increasing towards right side. You know, when we move on x-axis, the value of the variable increases. So this side, the concentration of nutrient is increasing. You can assume any nutrient, NPK, whatever you like, but concentration is increasing. Now you can see in the beginning there is short supply of nutrient in the plant or in soil, or low concentration of nutrient in the plant, or low availability of nutrient in the soil. But as you move, both are increasing this side. So in the beginning, it will be acute deficiency, when nutrient level either in plant or soil are too low to give good growth. So you can see the growth is hardly, say, 60% here. 60% growth is there. If you take it 100%, this Y scale is 100%, is starting from 0 to 100. Then with the lowest supply, you get less growth. But when the supply improves this side, you see the curve moves up. And in the next part, you can see it is latent or hidden deficiency. And in this zone, you find critical value. Critical value, as I already told you, it is the level of nutrient in soil or plant at which 90% of the maximum possible yield is there. Or you can say the level of nutrient in plant or soil where 10% reduction in yield is there, or we are getting 10% less yield. So that particular concentration in soil or in plant is critical level for deficiency. Critical level for deficiency. On the right side, last, you can see here, if it is 10% or more reduction, then it will be critical level for toxicity. Because here, it is not the deficiency, it is excess. Here up to this level, the nutrient, a particular nutrient may be in excess, and then it can reduce the yield. So the point where 10% reduction in yield or more is there, that particular point is critical concentration for toxicity in soil or in plant. So we started here acute deficiency. In the beginning, it was acute deficiency. Then comes the latent deficiency or hidden deficiency. So in this zone, plant is suffering from the availability of the nutrient or from the deficiency of the nutrient, but it may or may not show the deficiency symptoms. So if it is not showing the deficiency symptoms, but it still requires the nutrient, if you add the nutrient, the growth will occur. Then this particular level is your latent or hidden deficiency. So in this zone lies the critical value. Critical value is here in this zone. Then you still increase the supply. Then In this zone, you will get the optimal supply, where you are getting 100% of the growth or maximum possible growth in optimum supply. And then you still increase the supply of the nutrient in plant or soil, then luxury consumption happens. Luxury consumption means the nutrient is accumulated in excess, but it is not harmful to the plant. It will not affect the growth, but there is unnecessary use of nutrients at this level, luxury consumption. And then still you increase the concentration, then light toxicity can happen. This may be particularly for micronutrients like iron, manganese, copper, etc. So here toxicity can happen, and then further increase in a level of nutrient will cause acute toxicity. So you can see the growth is dependent upon the supply of the nutrient. There are deficient areas where you have shortage of nutrient. There are optimum supply zones and then toxicity zones. So we need to optimize. We need to run, or we need to have this optimal supply of nutrient where you get maximum yield, 100% yield with the optimum level of the nutrient. Now you can see the critical concentration. I just defined it. Critical concentration 10%. And in one of the lecture also we discussed about it. The critical concentration will vary with three things. Number one, the type of crop you have. Number two, the plant part which was sampled. Number three, the plant part which was sampled, and also the nutrient, which nutrient you have. For different nutrients, you have different critical level. For different plant, you have different critical level. And what plant part was used or sampled for critical level. And other is stage of the crop. At what stage? The critical level of nitrogen for rice, for example, during tilling stage will be different from maturity of flowering stage. So that you can see here. That you can see here. It is deficiency or toxicity. D is for deficiency, T is for toxicity. And critical concentration, you can see these are the values. And plant part analyzed. Which plant part was analyzed, and growth stage. So just let us see about nitrogen. Nitrogen deficiency, the critical level is 2.5%. Critical level means you need to go above this. Supply should go above this. And it was estimated in leaf blade at tilling stage. So you can see this critical concentration. Then C for phosphorus deficiency, 0.1% critical concentration, leaf blade, and at tilling stage. Now, if you see toxicity of phosphorus can happen, critical concentration is 1%. At least 1% is there, and it can be more than this. Then you will have toxicity of phosphorus in straw. In straw at maturity stage. So this is how you can see these, try to understand the critical concentration of nutrients. If you see about critical concentration in soil, for major nutrients and primary and secondary nutrients, we already have some rating chart that this is very low, this is low, medium, high, very high. We have those categories. But for micronutrients, we do not have those kind of categories. For micronutrients, either it is deficient or sufficient. So here, Katyal and Ratan have suggested critical limit in soil and plant tissues fully developed youngest leaf for micronutrient deficiencies in crops. So you can see for zinc, extractant that was used, it is mostly DTPA. For zinc, manganese, iron, copper. So we normally use DTPA as an extractant. So the critical concentration in soil is 0.6 ppm or microgram per gram, 2 ppm for manganese. Iron, it is 2.5 to 4.5 ppm. Copper, 0.2, boron, 0.5, molybdenum, 0.2. So these are the critical concentration in soil for micronutrients. And I suggest students to remember these values. These values are usually asked in your interviews and quizzes and so on. Then in plant tissues for zinc, 10 to 20 ppm, manganese 15 to 25 ppm, iron 50 ppm, copper 2 to 5 ppm, boron 5 to 30 ppm, and molybdenum very, very low. You can see 0.03 to 0.15 ppm. And for soil, for other nutrients, I think you know about available nitrogen 280 to 560, more than 560. These I have already discussed in one of the lectures. Now we have 5 different categories in India. Now, nutrient deficiency or toxicity symptoms. So when a deficiency will happen, whenever there is short supply of nutrient, you will get deficiency, and whenever there is excess supply of nutrient, then you will get toxicity. It is so simple. Range of nutrient supply from deficiency to toxicity. So you have seen these ranges. It is started from acute deficiency, then hidden hunger, then optimal supply, then light toxicity, and then acute toxicity. So these are 5 ranges starting from lowest to highest supply. The nutrient status of a plant can range from acute deficiency to acute toxicity. A broad division of nutrient status into 3 groups, namely deficient, optimal, and excess, may be useful for general purpose. Deficient, optimal, and excess. For a more accurate assessment of the nutritional status of plants, detailed categorization is required, in which 6 different ranges can be distinguished. So I have shown you all the 6 different ranges. One is acute toxicity, marginal or latent deficiency or hidden hunger, optimal supply, luxury supply, marginal or light toxicity, and then acute toxicity. So as you move from point 1 to 6, the level of nutrient increases in soil or in plant. Acute deficiency is associated with definite visible symptom. If acute deficiency is there, you will get definite visible symptoms or poor growth. It will surely reflect in this. So it is quite easy to judge the deficiency symptoms of a particular nutrient. Addition of a deficient nutrient results in increased growth or yields. Should be avoided, a sign of low nutrient supply and poor crop. So no farmer or nobody would accept this acute deficiency. You need to supply nutrients to correct this shortage. Then number 2 comes your marginal or latent deficiency, that is hidden hunger. A small range with or without visible deficiency symptoms. Here, in hidden hunger, you may have or may not have the deficiency symptoms. Growth and yields are definitely reduced. Addition of the yield limiting nutrient result in higher yield. Optimal nutrient supply prevents hidden hunger. So in this zone also, you need to apply the nutrients. Optimal supply range to aim for. All nutrients are at the optimum level or desired level. And here you will get the best growth, healthy green plants, good growth, and high yields with better quality. Range is generally wide for most nutrients. Optimal supply is reached above the critical concentration, which is generally associated with 90% of the maximum yield. Critical concentration serves as a diagnostic index for nutrient supply through plant analysis. Luxury supply, luxury consumption. It is useful to identify this range of unnecessary high nutrient supply. There may not be any negative effect on plant growth. Nutrient input is wasted and product quality as well as disease resistance may be reduced. That is the disadvantage. As such, it is not decreasing the growth, but some indirect effect like disease incidence or insect pest incidence may increase if you have luxury consumption of nutrients. It should be avoided. Optimal supply should be maintained and not exceeded. Can you tell me any nutrient which have luxury consumption? Anyone? The nutrient which is known for luxury consumption. With example. Hello?
Hello, sir. Sir, potassium.
Potassium. In which crop? In which crop? Crop. C-R-O-P, crop. So it is mostly related to potato crop. In potato, it is a starchy crop, and it also got high requirement for potassium. So it is, if it is in excess in the soil, then it can be accumulated in the potato, which is normally luxury consumption. So it should be avoided. Marginal, and then still you increase the supply of the nutrient, then you can have light toxicity, and which may be hidden. Here the nutrient concentration is moving towards toxicity above the critical toxic concentration. Crop growth or yield may have may be decreased, may be reduced, mainly because of toxic effect of certain elements or certain nutrients. And in this stage, there may or may not be symptoms because it is hidden toxicity. So can you tell me any nutrients which are toxic to the plant if their concentration is high? Can you tell me name of the nutrients which show toxicity to the plants if they are accumulated in excess? Any example? Hello? Any example?
Boron toxicity in cabbage, sir.
Yes. So actually this toxicity happens mainly with the micronutrient. One macronutrient is there, that is your phosphorus. Otherwise, N, K, or your calcium, magnesium, they do not cause any toxicity. It is mostly micronutrients like your iron, boron, zinc, even copper. So it is mostly related to micronutrients, this toxicity. Sometimes chloride toxicity can also be there. So next is your acute toxicity, very high concentration of certain nutrients, extreme supply, plants damaged by toxic level, toxicity, poor or no growth, poor yield, low quality damage to soil, plant health. The disease resistance of plants lowered and plant may die. This range should definitely be avoided for any nutrient. So this is commonly observed iron toxicity in case of rice, particularly in low pH soils or acidic soils. You may have iron toxicity. Of course, this aluminum is not essential nutrient, but still one can have aluminum toxicity in low pH or acidic soils. Now, nutrient mobility and its effect on deficiency symptoms. Yesterday, you have seen nutrient mobility in the soil. Now, you can see nutrient mobility and its effect on deficiency symptoms in soil. So, what happens? Redistribution of nutrients from original place of deposition is difficult for the immobile nutrients. Means the movement of immobile nutrient, as the name suggests, is less in the plant system compared to mobile nutrient. The movement of mobile nutrient is very fast and they can be redistributed. So their deficiency symptoms will come on older leaves. And in case of immobile nutrients, they do not move much in the plant system. They are not redistributed much. So therefore, their deficiency symptom will come in younger leaves or young tissues of the plants. So there are 5 types of deficiency symptoms or toxicity symptoms in the plant. If you remember these 5 types of symptoms, then you can very well explain the deficiency or toxicity symptom of any nutrient. Because in most of the cases, chlorosis occurs, whether it is iron, magnesium, or your nitrogen, sulfur. In many cases, you see the chlorosis occurs. So in many nutrient deficiencies or toxicity, chlorophyll is the first casualty. So chlorosis is very common to many nutrients. In some cases, necrosis is also there. Necrosis is death of plant tissues or sometimes in spots. Accumulation of anthocyanin resulting in purple or reddish color. So this is for phosphorus, but in case of potassium also, potassium deficiency in cotton, you can see discoloration. So discoloration is not always because of phosphorus. Sometimes potassium will also give you discoloration of leaves. Third one is lack of new growth, and then stunting or reduced growth. So actually it is very difficult to remember these deficiency symptoms for individual nutrient. So what happens is students can generalize that in most of the cases, the growth will be reduced, the color will change, the number of leaves, number of tillers, root growth. These are very general points. Of course, you need to remember specific points also. But when one or two nutrient deficiencies, toxicities are asked, you can have fair idea how to write it. If you remember these 5 points: chlorosis, necrosis, accumulation, lack of new growth, and stunting or reduced growth. All 5 are not applicable to every nutrient element, but they, if you keep them in mind, you can write very well. Then what are the factors for nutritional disorders in plant? So there are number of disorders uh, caused in plant due to short supply of the nutrients. They are not disease. They are not caused by microbes. Therefore, it is called as disorders. So what are the factors that affect the nutritional disorders? Means shortage or excess supply. So there may be soil factors. Soil factors can affect the nutritional disorders, means deficiency or toxicity of nutrients, like texture, because all these factors will affect the availability of nutrients. Availability and supply of nutrients will be affected by all these factors, like soil factors, texture, pH, organic matter, calcium carbonate content, type of clay mineral, soil moisture, temperature, climate. So these are the soil factors that actually affect the nutrient availability to the plants. Then crop factors are also important, whether a crop is able to take up the nutrients more efficiently or not. A crop or a variety which is not able to take a particular nutrient more efficiently, then it may show you some deficiency symptoms. So I have seen one variety of wheat that was very susceptible to manganese deficiency. It was not able to accumulate manganese. So likewise, there may be certain variety within a crop which uses a particular nutrient more efficiently than other. And so is the case with the crops. Certain crops can use one particular nutrient much better than another, like iron. Iron can be more efficiently used by wheat compared to rice. They are similar kind of crop. Iron can be removed by wheat as ferric iron, and rice likes ferrous iron. So rice suffers from iron deficiency, but this wheat does not suffer. So therefore, crop is also important. So crop factors are also important, like type of crop, variety, root system, and root exudates. Amount of root exudates released by the variety. They also help in the availability of the nutrients. So crop factors are important. Certain crops and variety may show disorder, but others may not show you. Then fertilizers, high analysis fertilizers are being used by farmers. So they are devoid of micronutrients. For example, long time back, farmers were using more of single superphosphate. Then it used to supply calcium as well as sulfur. And it also contained some minor quantities of boron also. So now the high analysis fertilizer do not contain those kind of nutrients. So it can be assigned as one of the reasons. Decreased use of organic manure, definitely, particularly micronutrients. If you use organic manures, then you will not require any micronutrient application. because they can supply the required quantity of micronutrients. Disposal of urban or industrial waste. Sometimes your soil gets polluted and it can affect the supply of certain nutrients. Lack of knowledge about balanced use. So these are the points that influence the nutritional disorders. Now you can see the mineral elements classified on the basis of nutrient mobility. So mobile, nitrogen, potassium, magnesium, phosphorus, chlorine, sodium, zinc, molybdenum. Of course, sodium is not essential nutrient. And immobile are calcium, sulfur, iron, boron, copper. So many times people put zinc in the immobile also, immobile category I have seen. But in case of zinc, the actually deficiency symptoms come first on the middle leaves, middle part of the plant, and then they may go up and they may go down also. So for zinc, it is not very fixed kind of tissues. You can say it is in between mobile and immobile, these two. Now, brief key to deficiency symptoms. So these are how one can remember these keys to deficiency symptoms appearing first on older leaves. Chlorosis is starting from leaf tips, then it is nitrogen. Chlorosis starting from leaf tip in the older leaves. Necrosis on leaf margins, potassium. Chlorosis mainly between veins, magnesium. Brown, grayish, white spots on serious, zinc, or chocolate brown spots. Reddish color or green leaves or stem, phosphorus. We'll see in detail also. Symptoms appearing first on younger leaves. Mottled yellow-green leaves with yellow veins, sulfur. Mottled yellow-green leaves with green veins, iron. Brownish black spots, manganese. Youngest leaf has white tip, copper. Youngest, so we'll see these symptoms at some other point of time. So we now we need to go to the